Hello, everyone. Hello, happy Wednesday. I hope you're all doing great. Thank you so much for joining us this evening. My name is Gabby, and I'm the Director of Education and Outreach here at FAU Harbor Branch Oceanographic Institute. Welcome to our John and Barbara Ferreira Ocean Science Lecture Series, the last one of the month. So we've had a great range of lectures this season. So happy to have you all with us. And I'd like to start by extending a very special thank you to everyone who supports our programs, including our John and Barbara Ferreira Ocean Science Lecture Series. We're very thankful for your contributions. And there are many great ways you can get involved to make an impact on the Institute, from volunteering your time, which we have many of you who submitted volunteer applications this season, so thank you, to sharing what you learned with your family and friends, and even making a gift to support our research and education programs. I also want to take a moment to thank all of the staff and, of course, the speakers that help with our series. This wouldn't be possible without them, and we have a great team. So as a reminder, this season, all of our lectures take place each week through March 26th. Your registration is good for the full season, so no need to sign up again, but we hope that you'll also encourage all of your friends and family to join us as well. And each presentation is also available virtually, so hello and welcome to everyone on our live stream. And if you miss a lecture, they're all recorded, so you can go back and watch them on our website. And for everyone here in person, remember after each lecture we host a meet and greet reception in the gallery with our speaker and some cookies, so please plan to join us for that. And also, if you'd like to pick up some flyers with information on our public programs and chat with Jen at our pop-up gift shop, we would love to have you do that. So we also hope you'll join us for next week's lecture on March 5th, and that's titled Coral Reef Connectivity and Conservation in a Changing Climate. And that's given by Ryan Eckert and Allison Klein, who are both graduate students working here on their doctoral degrees. So this will be a really great opportunity to learn from some early career scientists and also see how we're training the next generation of environmental leaders here at FAU Harbor Branch. And so this week, we're here to learn about aquaculture. Do we have any seafood lovers out there? Yes. So this is the presentation for you. Since you're interested in that topic, I have some other resources for you. So last spring, we hosted an Exploring Florida Aquaculture public workshop, and we had a variety of presentations from over 16 different people representing scientists, seafood industry, and more. And they talked all about some of the different plants and animals that were growing here throughout the state for food or for restoration. We also had a great session where we answered probably the question that we get the most: how do I pick out seafood that's safe for the environment or are good for my health? Which turns out is a 4-hour answer. But I'm saying this because all of these workshops were recorded and they're on the Harbor Branch YouTube page. So I love to tell people to go check out our virtual resources. And if you really want to take a deeper dive into aquaculture, we had a whole session on fish and a whole session on selecting seafood. And I think we did shellfish and restoration. So please find those there. And another great option is, if you haven't been there recently, I encourage you, or at all, to stop by our Ocean Discovery Visitor Center. We have a great aquaculture exhibit with some of the plants and animals that our scientists are culturing, and some other great information about the research happening here. And of course, every week on our tram tour of the campus, we drive through our aquaculture park if you'd like to see those facilities and learn more. And I'd be happy to talk to you about any of those options after the lecture. So, with that, the talk today is titled Seeds Are Only for Plants. Fish Farms Need Seedstock Too, by Dr. Paul Wills. So Paul grew up as an Air Force brat and lived in a variety of places throughout the United States, including inland and coastal states such as Hawaii and Louisiana. Paul has been a recreational angler and hunter throughout his life and gained an underlying love for the ocean and ocean life during part of his boyhood as he spent time in Hawaii fishing, snorkeling, and sailing, especially around the Pearl Harbor area adjacent to Hickam Air Force Base, almost on a daily basis. He began his aquaculture education at the University of Southwestern Louisiana, now University of Louisiana at Lafayette, studying crawfish culture, and completed his PhD at Southern Illinois University, studying the use of triploid hybrid sunfish as an option for management of sportfish ponds. After finishing his postdoc appointment at SIU, studying methods for production of all-female trophy muskellunge, he took a job operating a large fish farm that specializes in freshwater sportfish fingerling production for inland fishery management. In 2006, he and his wife of 31 years moved to Florida, and he has since risen to the ranks of full research professor and the associate director of research at FAU Harbor Branch. His primary areas of research today include fish production, new species development, land-based aquaculture system designs. And he has worked with a large number of species in his academic and private sector careers, including many species of marine and freshwater food fish and sport fish. So I'm really excited to hear all of Paul's updates today, and please join me in welcoming him.
Well, thank you, Gabby, for that wonderful introduction. And thank you all for coming and listening to me speak. So I'm going to talk to you about aquaculture, specifically about what we call finfish aquaculture, and how we attain the young animals for grow out. So aquaculture. So I'm going to start at the basics and try to bring all the boats up with the tide, so everybody understands as much as I can possibly get you to of my presentation. So aquaculture is the farming of freshwater and marine plants and animals. So I, in particular, work with finfish. So for those of you who are not familiar with that term, many of you may be, but some of you may not. We distinguish finfish from shellfish, basically. So finfish are those fish that swim. Shellfish are those sessile organisms, that sort of thing, in aquaculture. So I work with the guys that swim and can get away from you if you don't watch it. So why do we need to farm fish? This is a question that I'm asked often, but there's very good reasons that we ought to and need to farm fish. For one thing, from basic human nutrition, fish is extremely healthy to eat. It's very low-fat protein source, and it is typically very high in omega-3s. Even species that aren't naturally high in omega-3s in aquaculture, we can feed them diets, what we call finishing diets, that contain omega-3s, and that will boost the amount of omega-3 available to the consumer in those particular species. When we look at animal production, protein production overall, comparing pigs and cows and chickens, etc., in general, fish is going to be the most efficient from a resource perspective. So we actually use less water. You wouldn't think that. It doesn't seem sensical that we use less water for the production of fish than these other animals, but we do. And because we use systems that conserve water. The other thing is, if we look at commercial fisheries, oftentimes people look at the perspective of, while we have commercial fisheries, we can get seafood from commercial fisheries. However, the demand for fish today is outstripping our capacity for commercial fisheries to produce fish. So this is a graph produced and updated periodically by Food and Agriculture Organization of all the world's fisheries and the growth of the world's fisheries since, I think this is the 1950s. Sorry, I got to put my glasses. My wife hates it when I wear my readers. I'm of age though, right? So since the 1950s, which is the beginning of this graph, we see a large increase in production of commercial fisheries from our oceans. This is related to post-World War II intensification and industrialization of our commercial fisheries. And you can see that there's very little aquaculture going on at that time worldwide. But as we get towards the 1980s, you can see that commercial fisheries pretty well leveled off. So the productivity of commercial fisheries worldwide has leveled off. The interesting thing is a scientist that worked here at Harvard Branch named John Ryther back in the 70s, he was working at Hui. He made a prediction based on the total productivity capacity of the oceans and production of phytoplankton, that sort of thing, that the world's fisheries would plateau in the 1980s. And within about 10%, he was within about 10% of his prediction of where the world's fisheries would plateau and what level. And that is, in fact, where they've plateaued. So the ocean really has no capacity to increase productivity relative to what the demand is. And the demand gap is being filled by aquaculture. So aquaculture, oftentimes people who are proponents of commercial fisheries say, well, aquaculture is competing with commercial fisheries. In fact, it's not competing with commercial fisheries. It's filling the gap in demand that commercial fisheries cannot fill. So another aspect of this whole piece of the puzzle is sustainability of our commercial fisheries. If we look over time, this is from 1974 to 2021, which is when the statistics are available for, we see a reduction in the number of fisheries that are actually being fished sustainably. So more and more fisheries in the commercial sector are being fished unsustainably to try to push the demand. But you saw the previous graph, it's staying level. So that's not a good situation for the commercial fisheries. So these things, if we're going to have continued supply of good seafood available, we're going to have to change the paradigm, or we are changing the paradigm of seafood production. And we really are, the commercial, or the production of seafood is actually following, of course we're thousands of years behind the other products that we see in the stores. So the other products went from hunting and gathering to our traditional agriculture that we think of today. At some point in the past history of humans, that happened. That is what is happening now with our seafood production. So today, if we look at aquaculture, more than 50% of the seafood that we consume worldwide comes from aquaculture. And if we look at the U.S. specifically, only about 1% of that is being produced in the United States. So the United States is really falling behind in this production of seafood for our citizens. Currently in the U.S., we import over 80% of the seafood that we consume from other countries. We do export, wonderful. We do export, but when we look at the total imports versus exports, we have about a $19.5 billion trade deficit in seafood in the United States. We're actually doing better. In 2022, it was 24.8. So we've corrected a little bit, but we're at the point now where that graph of deficit is going up and down annually. So another aspect of fish farming that's often not talked about, but is very important, is the impact on local economies when fish farms are established. So if you establish a farm, there are going to be a lot of supporting industries that are required for that farm product. You're going to need to have seafood processors, folks that are buying the seafood and getting it to the ultimately to the markets. Then you also have folks like myself, researchers and other individuals that are improving the productive capacity of the farms and educating new people to work at the farms or to do research to help the farms. And then you also have all of the industries that supply components for the farms, feeds, et cetera, that the farms need. So one farm, when you start to add farms, you're adding all these other support industries and all these jobs associated with those support industries to the local economy. There are impediments to the growth of aquaculture that have been identified. One of the main ones is regulation. We hear that for a lot of industries in the United States. being a infant industry and coming into being in the U.S., we have a lot of oversight in aquaculture in siting and regulation of farms. The cost of feeds are always fluctuating and high. This is an area of a lot of research. Access to quality seed stock is essential for production. This is what we're talking about today. Disease control, societal acceptance of aquaculture products are also impediments. A lot of that due to myths about aquaculture. And access to capital for startups, for farms that are starting up. Because of the societal acceptance, oftentimes lenders are reticent to provide capital to new farms that want to start up. So some of the things that we can do, the requirements for successful aquaculture species to develop are to have high market value. So the species that we work with at Harbor Branch tend to be high market value species, and we'll talk a lot about pompano coming up, which we all know is a high market value species. The ability to develop captive seed stock for the fish, because without seed you don't have fish to grow. We like fish that grow fast, because fast growing fish grow efficiently. We want them to tolerate a range of temperatures and salinities and that sort of thing, because it means that particular species can be grown on a variety of farms. You don't have to have a real narrow set of conditions to produce those species. We will like for those fish to readily consume prepared diets. We don't want to have to harvest fish from the ocean or in order to feed them. We want to be able to do compounded diets that are highly nutritious to the fish. And we want them, of course, to be adaptable to a variety of different culture conditions. And I'll talk about different culture systems in a few slides. So if we're able to have a fish that will adapt to other variety of culture systems, then more farms can use those species. So seed stock. I thought we were growing fish, not corn, right? So why are we talking about seed stock? Because, like I pointed out in the previous two slides, the access to seed stock is critically important in order for aquaculturists to grow fish out so that they have fish to grow to market. They have to have the little guys, and that's what we're talking about when we're talking about the seed stock, or the small fish, the eggs that are fertilized and ready to grow or hatch and grow, the small fish, etc., that will go to grow-out operations. So when we look at aquaculture as a whole, we can look at aquaculture as a cycle. This cycle can be an annual cycle or a 2-year cycle, depending on the species they're working with. But you can say that it starts with brood stock, goes through hatchery production, nursery production. Those are what we call the seed stock components. And then you go to grow out. All right, so this is where the different systems, types of systems that I was talking about come into play, is that grow out component. If we look at the United States industry as a whole, these are statistics that come from the USDA statistics group. This is based on percentages based on number of farms, not productivity. Because if we looked at productivity, ponds would be the highest, because channel catfish is our highest productivity aquaculture species in the U.S., and they're grown in ponds. If we look, though, at total numbers, though, we have ponds. This represents our catfish industry in the United States. We have raceways. This represents our trout industry, which is our second highest productivity species being grown. And then we have recirculating systems, which are a fast-growing sector of aquaculture. So recirculating systems are systems that reuse water over and over again. They're always land-based. There are new pond systems that actually are a hybrid of recirculating technology and pond technology that the channel catfish is beginning to adopt. And they're seeing a huge bump in productivity, going from about 6,000 or 8,000 pounds per acre up to 10,000 to 15,000 pounds per acre by adopting that. Those would be still classified in the pond area, though, of this graph. But technically, they could be thought of as recirculating systems. But recirculating systems have tanks and filters, and the water is reused over and over again very efficiently. And then another large area that we see, or system that we see, are net pens. So most people, when they think about aquaculture nowadays, because of bad press and that sort of thing for aquaculture, think about net pens. But when you look at the total piece of the pie that actually represents aquaculture, net pens are a very small part of it. It's a very small part of it. But the rest of the aquaculture industry has to suffer for the bad press from that component, unfortunately. There's a lot of other systems that are very sustainable environmentally, and I'm not saying that net pens aren't environmentally sustainable. They can be operated in an environmentally sustainable manner, but you have to accept risk that goes with having those in the open ocean. And that's all I'm going to say about net pens. So the main thing that we're talking about today is seed stock. So we're talking about the brood stock component, which is your mamas and your papas that are going to produce the eggs, the fertilized eggs that you need for the further grow out of the different seed stock. So that the eggs go into the hatchery, they're hatched and they're grown, and I'll give you some details about that in a second. And then they go into nursery for further grow out to the size that the various different types of farms that I just mentioned would require. So different farms require different sizes of fish to go into their production cycles. So we'll talk about production of seed stock based on Florida pompano, which is a wonderful fish to work with. One of the things that sets seed stock production apart from grow out production is very complicated. There's a lot of different factors that are associated with risk. I'm not going to go through everything on this slide, but you can kind of see with all the different blocks and whatnot that are listed here, there's a lot of complexity to this component of the production system. It's going to be the most technically difficult component compared to grow out. Not saying that grow out is not technically difficult on its own, but the technical difficulties are primarily associated with maintaining good water quality, high quality feed, and managing the risks associated with growing for long periods of time. Whereas the seed stock production, your risks are related to all sorts of different effects that I've got listed up here. One of the unique things about what we do here at Harbor Branch is we use all recirculating systems. So much of what we do is applicable to what we call a flow-through system. So if you were to pump seawater through your systems, it would be very similar. But we want to demonstrate and develop technologies for farms that can be located inland where the land is less expensive rather than being tied to the seashore where land is very expensive. And because of that, these systems have to be very economical in water use, especially since most of the species we're working with are saltwater fish. So research system is a technology that we use and we develop at Harbor Branch for marine fish. So in a production cycle, we have our brood systems. This is a specific brood stock recirculating aquaculture system that has 2 tanks, egg collectors, and all the filtration that supports the fish in those 2 tanks. So this is what one of the tanks looks like in real life instead of on a drawing. You'll have the adults in that tank, and we can control the light cycle. So the 2 things primarily that control development of eggs and sperm that are competent for fertilization are light cycle and temperature. So we mimic the temperature and light cycle that occurs in nature, changing the amount of light concurrent with the temperature to mimic winter and spring, fall and summer, etc. The nice thing about it in a research system like this or in a system like this is that we can compress an annual cycle into a shorter period of time, where we can get 3 annual cycles, for instance, depending on the species we're working with, or 4 for some species, annual cycles within 1 year. That improves overall hatchery efficiency for seed stock production. So this is where we're conditioning the fish to be ready to spawn. So we have the thermal cycling, we have the light cycling, and then when we're ready to spawn, the system I showed is designed where we can divert the water into these collecting tanks that collect the eggs They have netting and that sort of thing, and oxygen, so that when the fish do spawn, the eggs go in there, they're collected, and then we can have access to those eggs. This is a system that we use where we allow the fish to spawn on their own. The term we use is volitional spawning. If you ever have been to a trout hatchery or something like that, you've probably heard of or seen strip spawning, where they actually take the fish that's ready to spawn and they'll strip the eggs out of the females and they'll strip the sperm and they'll mix them up, etc. So volitional spawning is the opposite of that. In volitional spawning, you get the fish conditioned, ready to spawn. They're in the tank together, the males and the females, and they do their natural behaviors and spawn and release the eggs in the tank. The water is diverted into these collecting tanks that allow us to then access those eggs. And it works well with these marine fish because they have eggs that are neutrally or positively buoyant that will flow with the water. Trout eggs are negatively buoyant, so it wouldn't work well with trout. That's why we do strip spawning with the trout as an example. So after the fish are conditioned, we will then check to see what the eggs look like, to see if a particular female or the group of females that's in the tank are nearly ready to spawn. This may have to be done a couple of times during the spawning cycle. And what we're looking for is to see whether a particular female within that group is showing signs of readiness. So we collect a biopsy of the ovary. It's very simple to do. You just pass this tube through the oviduct into the ovary, collect a small sample of the ovarian tissue. We'll put it on a microscope, measure the ova that we see there, and as long as the ova are above a certain size, we know that those ova are competent for fertilization. At that point, depending on the species, we might just manipulate the light or the temperature. Temperature, especially with red drum, when we do those, we just pulse the temperature, and that will induce them to go ahead and start spawning. With pompano, which I show here, we actually have to give them a hormone implant. So I want to emphasize something. The brood stock from a farm or from research and that sort of thing never find their way into the food system. And these hormones are natural hormones. It's a gonadotropin releasing hormone that stimulates the rest of the hormonal cascade that causes final maturation of the ova into eggs, and then ultimately stimulate the spawning behaviors by the females. The males, as soon as the females release pheromones into the water, and they're ready to go, and they'll spawn. That's us males, right? Don't tell my wife I said that, please. She gets mad at me about that stuff. So after they spawn, we collect the eggs. I just like this picture because it's a beautiful picture of eggs in it. But what I'm looking at here is probably, it looks to be about 30,000 eggs in that beaker. So the eggs are very small for pompano. And you can see they're a really pretty color, a really pretty straw color, right? And then if we look at them under a microscope, they look like this. So starting up here, we've got just spawned 0 hours post-fertilization, right? And then this is the sequence then of development of the egg. So we see real developmental landmarks. We see the layer of cells going across the face of the egg. So scientists and fish culturists, we want to see all these different stages, because we want to see that the eggs that we've collected are developing properly and healthy. And you can see by here, this time, you've got essentially a fully formed embryo in there. It's got a head with eyes. It's starting to develop a tail bud. And then finally, the tail is totally separate. The eyes are complex enough that you can make out the lens in the eye. The tail is extending longer. And then by the next morning, they're hatching. So it takes a little bit less than 24 hours for pompano to hatch at the temperatures that we culture them at. And then those larvae, these are the larval fish. In freshwater, remember a lot of my training is in freshwater. When I started working in marine systems, I had to totally change my terminology. In freshwater, we would call these yolk sac fry. In marine systems, we call these larvae. So they're the same. But you see the larvae, it has a tail, a head. At this point, it hasn't really developed its jaws yet. It is living off of its yolk. So the yolk is this large area here. You have an oil droplet, which is really a high energy packet for that larvae, and also helps it to float in the water column, because remember, oil floats on water, right? And then the other part of the yolk is amino acids and all sorts of other nutrients that young larvae needs to grow and continue development properly. So those are going to be found in what we call a larviculture system. Once again, it's a recirculating system. We use a technique called green water. When they get larger and are ready to eat, we'll green the water so that the larvae can actually see their prey. So these are larvae that are much more well developed. Here they're competent, they're swimming actively on their own. They have good competent eyes, they've developed their jaw, they have a rudimentary digestive system, and they're ready to eat. So what the greening of the water that we do allows them to have contrast, so that when a prey item, when a piece of food comes by them, they can see that food item better than they would if it was clear water. That's why we do the greening of the water. And we can use algae, we can use concentrated algae paste, or there's some greening products that are available. Some folks are starting to use kaolinite clays, which is the type of clay that they make fine china out of. But the idea is to have some particles in the water that provide contrast to that young fish so they can see their food item. So we developed these larval feeding protocols where the period of green water, we're going to be starting to feed a live feed item called a rotifer. This is a microcrustacean. It moves, it stimulates that fish to the predatory response in that fish. And then after they get a bit bigger, then we move on to a larger crustacean called Artemia. Those of you who are the men remember Boys Life magazine, and you could get sea monkeys in the Boys Life magazine. That's Artemia, by the way, sea monkeys. All right. And there's a huge industry worldwide for Artemia to support aquaculture of all sorts of species. One of the interesting things about both of these is they're not perfectly nutritious for the fish. They're lacking in some of the nutrients. So we go through a process of enrichment. We feed them what we call an enrichment product that contains the fatty acids and that sort of thing that they're missing, but the fish needs. So they become a living, swimming capsule with the nutrients in their belly that the fish actually needs. So that's a key breakthrough in marine aquaculture, by the way. I wish I developed it, but I didn't. I can just tell you about it. Once we get them large enough, we can shift them over to eating a prepared diet. We use very fine crumble diets. And this is a process that we call weaning. So we're going to wean them off of the live feed onto that prepared diet. And at that point, they're ready to go to various different grow outs. So live feeds is a very labor-intensive, resource-intensive system in marine aquaculture. And I'll talk about it later, but coming from freshwater where we don't do this, I would love to get rid of it. It works well, but it's a high cost for our culturists. So you'll see when I talk about our project later on, this is one of the areas that we want to reduce in the culture process, reduce or eliminate from the culture process of these marine fish. And it's not an easy thing to do. A lot of folks have tried, but we stand on their shoulders, of course, right? We know what they did. We stand on their shoulders, and we're going to carry that torch and see how far we can get. So at the end of weaning, we end up with a juvenile fish that looks like this, very different from that larval fish I showed you. It now has complete fins, even the spiny ray fins, basically the same finnage that it'll have when it's an adult. It has a competent eye, mouth, and digestive system, and is basically ready for independent life. Of course, in the wild, they're independent from the time the mother releases the eggs into the ocean currents.
How many days to get to that?
For us, this is about just right over 20 days, 22, 24 days. Depends on the temperature a bit. But our project is really reduced because when I first came to Harbor Branch, we were at about 34 days. So this is an area that we've really reduced getting to this stage. This is what a nursery system looks like. So that half gram, 1/3 to 1/2 gram fish would go into a nursery system for further grow out. And then from there, we would see the other seed stock sizes being 5 gram seed, or even growing to larger sizes to 50 to 100 gram seeds, depending on what a particular farm needed for its production cycle. Oftentimes, a farm will be set up where they can buy the smaller seed, do that grow out to 100 grams, that's then going to go into their production system, their final grow out production systems. Different farms will do things differently based on the economic calculations that they've done, of course. But the seed stock industry is designed to provide the sizes that a variety of different farms need. So in this case that I've shown you, they could provide fertilized eggs, they could provide those 1/2 gram juveniles, they could provide the 5 gram juvenile, or they could provide those 50 to 100 gram juveniles. So that's the seed stock that we're talking about. So we have a wonderful project. It's funded through the USDA ARS. We have USDA ARS scientists on site that work directly with us in our facilities. It's a collaborative research project between Harbor Branch and USDA ARS. It's called Seedstock Improvement to support the U.S. warm-water marine aquaculture industry. So initially we held a whole series of workshops to get input from industry, scientists, academia, et cetera, to determine what of these impediments that I showed you earlier we could address with a collaborative project like this. And after boiling down all of that, the information we collected, this was an area that we and USDA were interested in pursuing as a impediment. USDA ARS doesn't play in policy. Other agencies do that. So, USDA ARS's mission is basic science for application. So Seedstock was clearly an area that we could address with the type of project that we were setting up. It's a wonderful collaborative project, like I said. Along with our collaboration with USDA, we have multiple university and institutes that we've worked with. This project is in its 6th year. We've worked with many different institutes because, as much as we'd like to think that our scientists here at Harbor Branch are the best in the world, which I think we are, even the best scientists need help, right? So we reach out to partners to work with us because many hands make light work, right? The other part of the way we've structured this project is our industry partnerships. And to me, this is probably the most crucial of our partnerships. Having industry partnerships allows us to work directly with farmers. So in these 4 industries I've listed, 3 of these are farms. We do research on their farms. So not only do we collect real live data off of farms, but we also are instantly transferring the knowledge that we've gained from those experiments to the farmers. The other thing is it allows us to generate new questions that we can take to the lab and do under more controlled conditions, and then take what we've learned in the lab, bring it back to the farm, and do things under real-world conditions. That is the applicability key to the question, to the whole project. And I think this is one of the things that is really helping us with the success of what we're doing. Stratium is an industry that produces black soldier flies, which we'll talk about briefly in a little bit. It's a fly that can convert waste from vegetables and that sort of thing into a high quality protein. So the objectives of our project, this project, are to develop spawning strategies and larval culture methods. Now, most of the species we're working with have a lot of this done already. So for us, it's how do we make these processes more efficient so that the farms, when they're doing these processes, are more economically sustainable? How do we increase our understanding of fish nutrition and fish health? The fish nutrition piece is related to, we've got to get that from that half-gram fish up to 100-gram fish for the seed stock component. Luckily, when you get to that 100-gram size, what we find out nutritionally is applicable to the whole grow-out process. So that's transferability of that information. And then we also look at fish health. We want to have good, healthy fish that are being fed high-quality diets so that they're growing well. We want to develop methods for genetic improvement. So this is one of the aspects of aquaculture as a whole. There are very few species that we've done selective breeding with. I'm talking about the basic selective breeding like we've done with our dogs and cats and cows and pigs and chickens, etc., to have improvement. Here I am not talking about genetic modification or any of those other fancy techniques. I'm just talking about basic genetic selection for improvement. And with those well-known techniques that have been applied for in our beef industry for 16,000 years, we should be able to see huge improvements for the aquaculture industry. Of course, we have the ability to use genetic tools to see what we're doing at the same time to improve our speed of doing that. So it doesn't take us 16,000 years. I don't think it would be nice if they funded us that long, but I don't think they're going to. It would be nice though. So we've had a lot of, like I said, we've been working for six years so far. We've got a lot of accomplishments. I'm not going to list all of them. I'm going to list some of the key accomplishments the project has had. One is that we worked with the farms to develop techniques for low salinity culture in inland systems of larval fish. The reason this is very important is because if you can do the seed stock production in low salinities, that means that you don't have to spend as much money making full-strength seawater, right? And we found that we can get down to about 5 parts per thousand of salinity, which is about 14% seawater. That really improves the economics of production of the fish. We've reduced the requirements of live feeds. I said this is somewhere a piece that we want to eliminate ultimately if we can. We're at about 80% now after just the first 6 years of the project. And that's using diets that are already available from manufacturers. We have all the equipment and knowledge now using that as a baseline so that we can start tailoring those diets to pompano and redfish so that we can now improve that 80% number and try to get as close to 100% as possible. Another thing that we've done is we've determined oxygen requirements, which lowers the cost of production because farmers, when they do high density culture, need to buy oxygen. And if we can tell them what the limits are that they need to be at, now they don't have to buy as much oxygen to run their farms. Nutrition, we do a lot of work in nutrition. We look at new products that are replacements for fish meal. So fish meal traditionally is fish from the ocean, and we want to get away from using fish meal at all. With pompano, we've actually developed a zero fish meal diet that feed manufacturers could adopt if they were willing to or wanted to, which they will eventually. It takes time to go from research to application, but we've done that. We've looked at some byproducts from other fisheries, clam fishery particularly, looked at replacement of clam. We found that we could replace a lot of the fish meal in the diets with clam meal. This was with an industry partner out of New Jersey. We've worked with hemp meal using plant-based byproducts from hemp oil production. So after you've pressed the oil out of there, you end up with the protein components, and then you can make a meal out of that, which you can substitute for fish meal in the diets. We're doing a lot of work now with black soldier fly meal. Black soldier flies, like I said, are these flies that basically we're growing maggots and then drying the maggots and grinding it up into a meal. But it is converting a waste stream from basically old fruits and vegetables and that sort of thing. They can convert it into a very high quality protein. Fish eat maggots anyway. They love maggots, right? Anybody that fishes for bluegill has probably gone and bought waxworms before, right? So don't be disgusted by this. It is not disgusting. It is nature. But we can replace 25% of the fish meal in the diets of pompano or red drum with black soldier fly larvae meal. And we've shown that we don't have any issues with palatability to the fish. That's palatability to the fish, not the humans. We're actually farming a project out right now to actually look at the palatability to the humans, right? So that finishes the chain. Does using fish meal impart any adverse effects on the fillet from a nutritional or palatability standpoint to the consumer? I think this mic keeps going out on me, doesn't it? I'll try to talk as loud as I can. But regardless of the level of fish meal or black soldier fly meal we used, we saw good palatability to the animals. That was a very labor-intensive study because you have to hand feed the fish continuously and watch what they're doing. Are they taking the food? Are they spitting it out? And it takes a lot of time. It's probably one of the longer juvenile studies that we would ever do in our laboratory. So new feed ingredients to reduce fish meal and oil. We have another project that's funded by the Florida Specialty License Plate, where we do integrated multi-trophic aquaculture, which is a next generation recirculating aquaculture system. If you guys are interested in that, look at my last Ocean Science Lecture Series talk, and you can learn about integrated multi-trophic aquaculture. But one of the products we produce in there is a seaweed called ulva, and we can reduce that ulva into a meal. And we did substitution and found that we could substitute fish meal up to about 25% of the fish meal and effectively produce red drum with that ulva meal. So this is probably way in the future that we would use this as a product, but we know that it's a possibility. We do a lot of work to looking at nutritional requirements, various different carbohydrates. This is actually a huge breakthrough, determination of carbohydrate requirements of pompano. Pompano is a carnivorous species. They don't generally get carbohydrates in their diet. Oftentimes carbohydrates can cause problems in carnivorous species. So when I say Fernando, he's sitting over there, has determined the best carbohydrate source that we can use. And they can use it as energy, and they'll spare protein. So the protein can go to producing fillet, and they'll use the carbohydrate as energy. We want to determine methionine. These are essential amino acids. And then we've worked with other essential amino acids to determine how much a diet needs to have in them. So these are some of the different carbohydrates that Fernando looked at in looking at the carbohydrate sources. He covered the gamut of different ingredients that are being used by feed manufacturers. Broodstock, like I said earlier, is a very important component. We've established spawning groups of multiple different marine species that were regularly spawning. This allows us to have young stock for doing these nutrition studies and other studies. We've got established methods for out-of-season spawning of the California yellowtail. So we have partners in California. This project is a nationwide project. It's not just a Florida project. So we've got partners in Texas and California and Florida, et cetera. We've looked at nutritional requirements for the broodstock themselves, not just when we're growing them, but how do we maintain and ensure that we're going to have good quality eggs coming from the broodstock. Looked at the nutritional link between the eggs and the broodstock for red drum pompano and California yellowtail. Established the required amount of time to change the nutrient profile of California yellowtail eggs. So this is important. So we haven't done this with red drum and pompano, because with red drum and pompano, when we spawn them, we give them the hormone or pulse the temperature. They spawn, and they spawn all the eggs that they've got. California yellowtail, on the other hand, when you stimulate them to start spawning, they'll continue to spawn over and over and over again periodically. So they're a fractional batch spawner rather than a synchronous batch spawner. And they're going to be continuing over the course of weeks and weeks and weeks, producing batches of eggs for the farm. So what is the effect? We've looked at the effect of the nutrient profile over the time if we change the nutrients. How long does it take if we're feeding that fish a specific diet for the profile of the eggs to change to reflect what's in the diet, which is critical to management of a broodstock population for good quality eggs for that species. In genetics, we have sequenced and published the sequence of the pompano genome. So we know what all the AT, Gs, and Cs are, and we have completed a draft transcriptome to the scientists in the audience. They know that means we know where a lot of the critical genes are for that fish. We've looked at the molecular response of larval pompano. So this is important genetically, but it's also important in that when we're looking at developing diets for those young fish, because we want to know what genes are being turned on when and how they're using the nutrients that they're getting, how their gut microbiome is developing, et cetera. We've currently finished collecting samples and are sequencing so that we can look for genetic markers within this sequence, within this genome, looking at genes responsible for growth. Head size, you say, why would we look at head size? That's weird. We look at head size because if you can reduce the size of the head without affecting their ability to consume the food, et cetera, and swim around in the tank, you're producing a bigger fillet, right? So this is critical, not as critical with pompano, but is critical with red drum because red drum have a very large head compared to their body. And we're not just doing this on a whim because we think it's a great thing and it really sounds cool. This has already been done with striped bass in aquaculture, and we know that it's something that we can affect, and we can increase the product, what we call the dress out percentage of that animal, which means more revenue to the farm for the animals that they harvest. One of our farm producers was seeing a sparkly silver coloration in some of their fish that they were growing, and they were concerned that this might affect the consumer appeal for their product. So we did a quick study to get samples of fish that were showing this trait and fish that were not showing this trait so that we can see how it's genetically controlled. So a genetic selection program, a standard genetic selection program is time consuming and quite difficult. But since we have intimate knowledge now of the genetic sequence of these fish, we can look for markers for good growth genes and that sort of thing. And we can use a new process called marker-assisted selection. So instead of just looking at a fish and saying that fish grew faster than all of its compatriots in the tank without knowing exactly what's happening genetically, we can take a fin clip from that fish, which is a non-destructive sampling technique. We can sequence that and identify markers for good growth and that sort of thing and say, this fish has the markers for fast growth, or this fish does not. Even though it grew fast for some reason, maybe it was a hog or something, I don't know, if we have known markers, we can go into broodstock and we can select out the particular males, dams and sires, males and females, that we want to do paired matings of to get improvement, rather than mass selecting groups of fish that show traits. So this will vastly increase our ability to change. Another thing that we were interested in is how much genetic variability are in the populations that are available to us for the different species. So we've gotten fish from the East Coast, West Coast of Florida for pompano and red drum, and looked at how much variability and how much overlap between the populations there are, because that will define where do you select your fish from and that sort of thing. And we found with pompano, there's almost no difference between East Coast of Florida and Gulf Coast pompano, and very little. And we were very surprised by this, because the general consensus is that there are big genetic differences in red drum, but the data that we're collecting is showing just the opposite of that it's more like pompano with very little genetic variability between the East Coast and West Coast of Florida. Another key aspect of what we do is outreach, getting this information to farmers as quickly as possible. We've developed a culture manual for pompano so that farmers basically can hopefully pick up our manual and start a farm. They would obviously have to have some other knowledge, but this is basically a roadmap of how to do everything that we know how to do. We didn't publish this as a paper manual. We've kept it as an electronic manual because we want to continuously keep it updated. As we learn things, this manual will have different versions that incorporate what we're learning so that we can get that information to farmers as quickly as possible. It's a very comprehensive manual. We've got workshops that we do periodically, annually. We try to record all the talks that we have at those workshops so that they're available in perpetuity on our Harbor Branch website. These are both workshops that are done here, like the previous slide, and workshops that are done nationally. And then we are always planning for the future. Why did it do that? Okay, we're always planning for the future. So we're building a disease challenge facility, which will allow us to do selection for individuals that are resistant to disease. This is what that facility looks like. It's about 50% done now. We just got our fire marshal inspection, which is a big deal for anybody who has ever built a building knows. That means we can occupy the building and actually start putting stuff into it. We're building a selective breeding facility, or expanding our capabilities in our selective breeding facility and our broodstock maturation facilities. Those two go hand in hand. We're developing a brand new, this is groundbreaking work that we're doing, rapid on-farm nutrient detection system. We've purchased a new device called a Raman spectroscope, which will allow us to go to a farm, collect a batch of eggs, put this in the test tube, and it'll tell us what the fatty acid profile is, the amino acid profile of those eggs. And with the knowledge that we know of what we're looking for in good quality eggs, now we can make an assessment on farm within just a few minutes of the quality of those eggs, and we can help the farmer then to make changes to their broodstock diet so that they're producing good quality eggs. And then the feed production lab, we have an experimental extruder, which will allow us to make diets that are similar to the diets that farms use. It'll allow us to do experimentation on production of diets. That data can go then directly to diet manufacturers so that then they can make those diets for the farms. And then we do all of our education. We have a lot of wonderful students in our program, graduate students, master's, PhD. Here we have Jasmine. She was an undergraduate that's working with us. And you can see she gave this presentation, won the top prize for her presentation. So this is to me, as an educator, is one of the most wonderful parts of this project. This and working with farmers, of course. Being a farmer, I loved working with farmers. So, and then being an educator, I love working with students. So That's all I've got for you, except for questions. Any questions? I've got one.
In the beaker, you have 30,000 eggs. How many fish does that represent?
Well,
How many fish does it take to produce 30,000?
so the question is, in a beaker with 30,000 fertilized eggs, how many fish does that represent? So if everything was perfect, it would represent 30,000 little fish. Everything's not perfect. I showed you that graph of all the things that can go wrong, right? So we realized about 50% from that. Yeah.
And how many fish produce those 30,000 eggs?
One female can easily produce that many eggs. Yeah. One female can produce upwards of 100 to 300,000 eggs, depending on her size. Yeah. So that was a really small spawn, or only a really small part of a larger spawn, right? That I showed in that picture.
Why pompano?
Why pompano is the question. Pompano is probably the most delicious fish there's ever. Even Mark Twain, there's a famous quote by Mark Twain, where he compares eating pompano to the greatest of sins. I don't remember the exact quote, but basically that's it in a nutshell. It's really a good fish. For folks that don't like oily fishes, oftentimes they still like pompano because the flavor is so good. It's actually it's an oily fish. It's a jack, and people think about crevalle jack and say, oh, yuck, crevalle jack. But pompano is on the total opposite spectrum of acceptability. And it fetches a very high price at market. So I haven't gone and looked at pompano. I don't buy pompano. I can't afford it. But I grow pompano, and that's how I get my pompano, right? But in the market, to a fillet, and the retail market, it's going to be about $23 to $25 a pound. So the idea is you have a high-value fish the farm can get well established with good profitability, and then a profitable farm can start to diversify into other species, which is good for the farm. Diversity is always good for the farm. Diversified products, that is good for a farm because they can be producing products more and they have more income streams coming into that farm. But the key to a startup is good profitability. So that's one of the reasons for pompano, among many others. It works. It meets a lot of the criteria that I showed. It accepts a pelleted diet. It works well in multiple different types of systems. It's very resistant to water quality, and it is a fish that moves a lot, so it burns a lot of energy. So you have to have very well designed diets for it. Have I personally in my career? Yes. Bluegill. Bluegill are wonderful fish. They taste great and they're wonderful fish. They produce a wonderful fillet, but it's really hard to get them large enough to go to market. And with the imports of tilapia, most people will buy tilapia because it looks just like a bluegill. And in fact, a lot of times in the Midwest, you'll see tilapia sold as bluegill. Down here, you'll see tilapia sold as grouper and everything else, but just so you know.
Question about pompano. Why is there such a big difference between the cost of pompano that's bought in pompano? I was at a fish market in Pompano about 2 weeks ago. The fish bought in Pompano was $20-some a pound. I go to this fish store and it was $6 a pound. So I asked the guy, what's the difference? He said,
So it's probably, so the difference between the pompano that you saw that came from China is probably blunt nose pompano or the the golden pompano or one of those species, is that those countries highly subsidize their farms and we do not. Yeah.
But are they equal? They're the same in terms of flavor, taste, and all of that?
I think our pompano are better. The other thing about our pompano, if you buy a pompano produced in the United States, you know that we have regulations, It's going to be grown with a high quality diet. It's not going to be grown with a lot of antibiotics. It's not going to be grown with any bad chemicals, and there's very little regulation in other countries. So when they test, they do detect these, chemicals, et cetera, in the fish and antibiotics in the fish coming to the U.S. So you can be assured if it's grown in the U.S., it's grown under a more highly regulated system, and it's probably going to be a lot fresher. That fish that was grown in China is going to have been frozen. There's no telling once it's frozen how long it sat on a boat on the way over here. It would have been frozen the whole time, but the longer you freeze oily fish, the more even you'll still get rancidification of the oils. So you're going to get a higher quality, fresher product if you're buying U.S. pompano. She's been trying to ask a question. I keep pointing at her.
Well, I remember tasting a wonderful rainbow trout from Seedstock, California, and it was from a farm called McFarland Farms. And the interesting thing about that trout is that it was fed algae pellets. And I was wondering, with pompano, how much of the diet is a plant-based diet?
So when I talked about the diet where we were able to eliminate fish meal products, so most of that was soybeans and other grain meals to make up most of the protein. But the oils are another component. So people often talk about fish meal, but the fish oils is also an important component of the diets. And in that diet, we fully substituted all the fish oil as well, which is probably a bigger breakthrough than substituting the fish meal. And the oil that we used in that study was called Vermeris oil, which is a algae-based oil. So as far as substitution of algae for pompano production, we haven't done that study yet. It is on our plan. Very much like I showed you the study with the red drum, right? So we have plans to do that same type of study now with pompano as well. So I don't have the answer for you today, but check back in 4 years. Here and then there. So you first and then you.
Farms that are growing a high concentration of fish, is it a pollution problem?
So in recirculating aquaculture, no. You have complete control over everything coming into and off of your farm in recirculating aquaculture. In pond culture, very little, because most of your water loss is to evaporation and it's primarily fresh water. So you're bringing water into the farm. Most of what you're losing is to evaporation and seepage. And when we look at catfish farms, the acreage of the ponds is so large, 10 to 20 acres, that it's just not economical for them to drain those ponds. And draining the ponds would be the only time that the pollutants could get out of the pond. So it's not economical for them to drain those ponds frequently, so they don't. The place that we think about pollution would be in flow-through raceway culture or flow-through tank culture, which is primarily done with trout. But nowadays, with the new regulations that are in place, they have to have complete treatment of the water before it's discharged into the receiving stream. So usually you have a stream with a nice cataract spring that feeds into that farm, and then the water goes through the farm and then discharged into the stream. That's the way it used to be done 30 years ago, 20 years ago. It's not done that way anymore. They do strict monitoring of the outflow of those farms. If they go even a scintilla over the phosphorus export that they're allowed, they'll shut that farm down. So net pens, if they're operated properly, it shouldn't be. That's all related to siting and that sort of thing. So you have good flow that moves those nutrients away from the pen. Integrated multi-trophic aquaculture is actually a system that is used For net pens, you locate seaweeds and mussels and that sort of thing around the pen to capture the nutrients from the plume that's coming off, because you try to locate where there are currents. So that's a way of mitigating that. Ultimately, though, what generally happens is you start supporting a new ecosystem around the net pens. And in fact, historically, there have been experimental net pens that have been put in Hawaii, and there was a lot of backlash when they were getting the permits for those experimental cages, particularly from recreational fishing and the charter fishing industries. They thought they were going to ruin their fishing. Ultimately, after years of operation of those experimental net pens, the experimental permits expired, and they were removing those net pens. And then they got pushed back again from the same group, because those net pens became such concentrators of fish that the industries were really reliant on them to ensure that their customers were catching fish. And so, nature has a way. Now, we don't want to overwhelm the systems, of course. And like I showed you, there are very few farms in the U.S. that are permitted for offshore aquaculture. Now, if you look at the way it's done, say in China and some of these other Indo-Chinese countries where the regulations are much lower than we have, the siting is not proper, those sorts of, and you can get, and so the U.S. aquaculture industry suffers from the bad image that's been brought forth because of bad actors in other countries. But it's okay. We want to have a well-regulated industry that are good husbands of our husbands in the sense of husbandry of our ecosystem, right? So a little bit of regulation is good to ensure that we do things properly. I promised her next.
How often do you try and go back, especially with the food changes that you made during the aquaculture process, to assure that the fillet you're achieving tastes like the wild caught?
Okay, so this is a very good question. The question is, how do we ensure that the fillet that we produce is just like the fillet that comes from the wild? In taste. Never going to happen. Because, yeah, everybody wants to volunteer. It's the whole story of Henny Penny, right? And I'm going to now explain my answer because it's not bad. So a wild fish in the wild has a varied diet. It's basically eating what it can within the scope of its ability to digest and capture and that sort of thing. But if you look at a pompano, it's going to get crabs, it's going to get sand fleas, it's going to get, other crustaceans. So it's getting a whole variety of different fatty acids and different proteins and that sort of thing as it's growing up from that little fish up to the large fish that we struggle to catch in the surf. Especially right now when we have so much algae out there in the surf. I was out there yesterday. I didn't catch a darn thing except a bunch of algae. But so that's a wild fish, right? It's got a whole bouquet of different flavors that it's accumulating throughout its lifetime. In aquaculture, it's not economical to do that. We ensure that our diets have very good fatty acids, very good proteins, highly digestible. So one of the myths in aquaculture that many of you have probably heard is that, they feed poop to their fish. I'm sure if you can raise your hand, you've probably all heard this myth. It's a myth. It's not the truth. It doesn't even make sense if you think about it. Because if you're a farmer, you want to grow healthy animals. You want to grow healthy animals that are going to produce a high-quality product that you can sell and support your family. You're not growing them to feed your family. You're growing them to get cash so that you can support your kids and send them to the best schools, et cetera, et cetera. If you're feeding poor-quality feed to those animals, you're not achieving your mission. So it doesn't even make sense for somebody to say that. I know probably where the myth comes from. It's a long, detailed story. I'm not going to tell it, but believe me, it's a myth. So we produce, or the fish are fed a very high quality diet that's going to have the amino acids and the lipids, the fatty acids, especially the omega-3s, et cetera, that are heart healthy. And the flavor for 90% of consumers, they'll never tell the difference. It's only the most sophisticated of consumers that would be able to detect the difference. And in fact, we worry more about what we call off flavors. So in a standard recirculating system, you get bacteria and that sort of thing that produce these compounds called methyl isoborneol and geosmin, which can impart a musty or earthy flavor to the fish. That flavor is great if you're growing mushrooms, not great if you're growing a fish. So if you have mushrooms, you want those flavors. If you're growing fish, people don't want that flavor. So we do a lot of things in purging the fish at the end of the production cycles, ensuring that those compounds aren't being produced, et cetera, in order to reduce those off flavors. And those flavors are the flavors that most people will complain about. And here in Florida, most people will say that fish tastes like a freshwater fish, because most of the freshwater fish you catch in Florida, they're exposed to a lot of blue-green algae in the fresh water, which impart geosmin to the fish. So he's been raising his hand.
Could you just tell us a little bit about the red drum? Are they raised for food in Florida? Or are they being raised for sport fishermen, finger lakes, to reproduce wild stuff?
So the state of Florida has a program for production for stock enhancement. How they don't produce, they're in the process of getting to where they can produce. They're building some hatcheries and that sort of thing. I'm not sure that they're doing any stocking yet. Years ago, 2008, 2009, we did a bunch of work for them, developing recirculating technology for production of juvenile red drum for stock enhancement. Luckily, all of that work also plays into this project, because it's the same process going for stocking fish that you stock and fish that are seed stock for grow out. The genetics is different because you want to represent the natural genetics if you're doing stock enhancement. With aquaculture, you want to have select fish that grow faster, which doesn't mesh with stock enhancement. You want to represent the natural gene pool if you're doing stock enhancement. So there is stock enhancement production of redfish. It's primarily going to be by state agencies. The production of redfish for food, there are some farms that are starting to come online. One in Pine Island. There's a farm up in the Pensacola area that's working on development, which incidentally would be a net pen farm. The one in Pine Island will be a recirculating farm. So you can judge whether one is sustainable or not. That the jury's still out on net pen. But they will be being produced in the state of Florida, and it is legal to produce them with the proper permitting. So a lot of people think it's illegal to produce redfish, but the state law only restricts largemouth bass and snook. And about 2 or 3 years ago, the rules were changed for largemouth bass. So you can do largemouth bass production in the state of Florida now, but you have to have a complete genetic surveillance program in order to do it for your brood fish, to ensure that your brood fish are all well, it's now the Florida species. It used to be the Florida subspecies of largemouth bass. That you have no genes from the northern largemouth bass. So.
All right, everyone. Let's thank Paul for his excellent presentation. And if you all have additional questions or want to continue our discussion, please join us in the gallery. We'll chat with Paul while we're eating some cookies. And we look forward to seeing you all next week for our first lecture in March on coral reef connectivity from 2 of our graduate students. Thank you all. Have a great week.